All right, let's dive into something pretty incredible today. We're looking at a brand new way to find cancer's biggest weaknesses. It's a platform from a company called Gordion Biosciences, and their whole goal is to crack cancer's code by looking at the one thing that truly matters, real tumors from actual patients. So let's see how they're doing it.
But before we get into the how, we have to start with a big and honestly a really frustrating question. Why is it that so many drugs that look amazing in the lab just fizzle out when they get to human trials? It's a massive roadblock in oncology.
And a huge reason for all that failure really comes down to two simple words: false positives. The old school ways we hunt for cancer drug targets. Well, it turns out they're often accidentally pointing us in the complete wrong direction. They're finding weaknesses that just aren't the real deal in actual people. Okay, so let's really dig into this whole false positive problem. Let's talk about how the traditional methods have been, leaving us astray for so long. So, on one hand, you've got the traditional path. This usually means using powerful tools like CRISPR on cancer cells grown in a petri dish. Now, these tools are amazing, but those lab-grown cells, they're not perfect copies of what's happening inside a human body. Gordion's approach, it's a total shift. It goes right to the source. We're talking thousands of real patient tumor genomes to let the tumor's own story of evolution tell us what it truly needs to survive. So this new way of thinking, it's like treating a tumor's genome as its own personal diary, a super detailed record of its long and brutal fight for survival. And the real key, of course, is knowing how to read it. The author of this diary, if you will, is this concept called selective pressure. See, as a tumor grows, it's constantly being forced to adapt. It has to keep what it absolutely needs to live, and it's happy to toss out anything it doesn't. The most important part? This whole process leaves behind a trail of clues. And the rules of this evolutionary game are actually surprisingly simple. If a gene is absolutely critical for that tumor's survival, the tumor's going to guard it with its life. You almost never see that gene get deleted. But if a gene is non-essential, or maybe it even gets in the way of growth, the tumor will dump it without a second thought. It's literally evolution playing out in real time. So how do you actually read this diary and apply these rules? Well, you need a kind of genomic detective kit. And for Gordion, that kit has two main things: a massive pile of evidence and a really, really smart magnifying glass. First up, the evidence. And listen, we are not talking about a handful of experiments in a lab. We are talking about analyzing the genomes from over 9,000, almost 10,000 real patient tumors, spanning dozens of different cancer types. That kind of scale is how you start to see the real patterns emerge from all the noise. But here's the thing, just having a mountain of data isn't enough. A lot of the publicly available data is processed in different ways, and that can hide really important clues. So Gordion reprocesses everything through one single uniform pipeline. It's kind of like cleaning all the evidence from a dozen different crime scenes to make sure every single fingerprint and fiber can be compared fairly. And here is that magnifying glass I mentioned. It's a proprietary AI called the Functional Copy Profiler. It basically does three things. First, it pulls together all the genetic data for a tumor, mutations, copy numbers, the whole shebang. Second, it accounts for all the weird specific things about that tumor, like its chromosome count or what scientists call ploidy. And finally, and this is the most important part, it counts the exact number of working gene copies left. That difference between just any copy and a functional copy, well, it's everything. And get this, the tool is incredibly precise. When they tested it against benchmark datasets, it hit about 99% accuracy. That is the kind of reliability you absolutely need when the stakes are this high. So, with this super accurate tool in hand, Gordion was able to spot a critical clue, a key piece of the puzzle that most other methods just completely overlook. And that clue has a name: partial loss, or PLS for short. Now, this isn't about a gene being totally wiped out. No, it's the much more subtle event of losing just one single functional copy of a gene, relative to how many chromosomes the tumor has in total. And that distinction, it changes everything. You see, a complete loss, where both copies of a gene are gone, that's actually pretty rare for most genes. But partial loss, that happens all the time. And by focusing on that much more frequent signal, we get a way clearer picture of what the tumor is really trying to protect. This chart just lays it all out perfectly. When you look across all those thousands of tumors, a crystal clear pattern just pops out. Genes that are truly essential are protected. You can see partial loss is low. But for non-essential genes, partial loss happens a lot. The bar is high. The tumor's diary is basically screaming at us, telling us exactly what it can't live without. All right, so we've got the data, we've got the tool, and we've found the key clue. The final step is to put all of this together and create a final verdict, basically a ranked list of cancer's biggest weaknesses. And that verdict is called the Synthetic Lethality Index, or SL Index. It takes all that incredibly complex biology we've been talking about and boils it down to a single score. The logic couldn't be simpler. The lower the score, the more essential that gene is for the tumor's survival. This lets researchers zero in on the absolute best drug targets, the ones with the highest chance of actually working. And all of this leaves us with a final, really powerful question. By learning to read the evolutionary history written inside thousands of real patient genomes, we're getting closer than ever to a new reality in cancer treatment. What if we could finally move beyond all the false positives of the past and find the precise, validated weakness in every single tumor? That right there is the promise of this whole new approach.